Welcome back, dear students, to one more session of genetics. Today we are going to talk about crossing over. What is crossing over? Crossing over is a process of reciprocal exchange of genes between chromosomes of a homologous pair, resulting in their changed constitution. Now, what do you mean by homologous pair? A homologous pair refers towards the chromosomes which contain the same genes. If you go to consider a human, we have that in the 21st chromosome, we have two copies of the 21st chromosome, as well as every other chromosome, we have a pair. Okay, so if you go to consider that pair of chromosomes which contain the same uh, what the same type, the genes coding for the same genes, they are referred to as the homologous chromosomes. And these homologous chromosomes, one is being derived from the paternal member of the person, of the individual, and one chromosome is derived from the maternal partner. That is, one is derived from the father as one is derived from the mother. So if you go to take every individual, he has a pair of chromosome which has been derived from his father, and one is being derived from his mother. So the homologous pair of chromosome refers towards the chromosomes which contain the same genes, and the mutual exchange of or mutual interchange of chromosomal parts between non-sister chromatids of a homologous pair of chromosomes, resulting in the recombination of genes, is referred to as crossing over. And as a result of crossing over, new combinations of genes or recombination will occur, and crossing over is an important source of variation among the sexually reproducing organisms. And the gametes which have been formed as a result of crossing over are called the crossovers, and those without crossing over are referred to as non-crossovers. Now, if you go to take every individual, their offsprings might be sometimes having some variation from them, or in some cases you can find that the offsprings will be found to be very identical towards the parent. So, if the offspring is being found to be very identical to the parent, it indicates that crossing over has not happened. That is, recombination has not been formed when the gametes involved in the formation of that offspring was being formed. Such cases would lead into a case where what? Not much variation is there. That is, if the offspring is very identical to the father, or not much of recombination or crossing over has been happened. But if there is a variation from the father or the mother or from the parent, generally speaking, it indicates that crossing over or variation has been happened. Now, if you're going to take every individual, now consider this is a individual, the father, you consider the father, okay, and this individual, now if you're going to take here, you're taking Will Smith, okay. Consider Will Smith as such. Will Smith is having a chromosome which he has obtained from his father, and one which he has obtained from his mother. And when that individual is producing his gametes, okay, what happens? The chromosomes, the homologous chromosomes, will pair with each other, and they will synthesize double the amount, or a duplicate or a sister chromatid is also being synthesized during the process of meiosis. And these homologous chromosomes might sometimes just undergo the process of meiosis without undergoing crossing over. You just think the exchange of genetic material, consider it is not happening over here. In such cases, all the gametes which have been formed here would be what? Identical to the what? Identical to the parent over here. In such cases, these gametes are not they are not undergoing crossing over when the gametes are being formed, or the gametes of which are being formed here are being found to be identical to that of the parent over here. But sometimes when these gametes will be formed, what happens? The maternal and the paternal chromosome of the individual of this parent would undergo some chromosome crossing over or exchange of genetic material, and as a result sometimes, now here the gamete is containing only the paternal chromosome, and in the case of here it only contains the maternal chromosome, but here you can see that some amount of crossing over has been happened. And so depending upon whether crossing over has happened or not, or the extent to which the crossing over has been happened, the gametes will undergo variation during the process of meiosis. Oh, hope so you know the process of meiosis, that I'm not going into the much details of it, but you can see that meiosis mainly consists of two stages, is meiosis 1 and meiosis 2. And in meiosis 1 itself we are having prophase, metaphase, anaphase, and telophase and cytokinesis. And in meiosis 2, we have prophase 2, metaphase 2, anaphase 2, and telophase followed by cytokinesis. And the process of crossing over, it mainly happens within the prophase 1 stage of meiosis 1. If you go to consider the steps of crossing over, you can see that in the prophase 1, there is a stage called the zygoten stage, where what happens, the homologous pair of chromosomes, they move towards each other and they start pairing. Okay, so the pairing process is referred to as synapsis, and each homologous chromosome of the bivalent will split longitudinally to form two identical sister chromatids and will result in the formation of four chromosomes. This is called the tetrad stage. I will be showing a pictorial representation so that it is more clear to you. And the crossing over, it involves the breakage and exchange of parts of, as well as the exchange of parts between two non-sister chromatids of the homologous pair of chromosomes, and this process, an X-like structure at the point of exchange of chromatid segments, or the point where the crossover happens, is referred to as the chiasma. And after the completion of crossing over, the non-sister chromatids will repel each other and they separate from each other from the centromere towards the chiasma and to the end of the tetrad, and the separation is referred to as terminalization. And once terminalization is brought about, the homologous pair of chromosomes get separated, and you can see that each event of the crossing over produces two recombinant chromatids called the crossover chromatids and two non-crossover chromatids or the original chromosomes. So if you go to consider the different stages pictorially, in the prophase 1 you have different stages, leptoten stage, zygoten stage, packaging stage, diplotin stage, and the diakinesis where the terminalization occurs, and then you have the metaphase 1. So you just remember, if you're going to consider meiosis 1, in meiosis 1 you have a prophase 1. This prophase 1 itself is containing different stages. That is the leptoten, zygoten, packaging, diplotin, and diakinesis. We'll be going in the details of this during the process of cell cycle, but at this zygoten stage, okay, now you just look, the chromosomes are being found to be in leptoten stage. What happens? The chromosomes are being synthesized here, and what happens when it comes to zygoten stage, they are being found, the homologous chromosomes will be found to be paired with each other, and they are being aligning with each other, and the complete pairing happens in the in the packaging stage. And what happens, if you go to look into this packaging, I can just show you a picture where, okay, now here this is in the meiosis 1 they are entering. Now you have a duplex is being formed, okay, and see, in this chromosome also a duplex is being formed, and when they pair together it will be resulting in the formation of a tetrad. That is, you can see this tetrad over here, and then in this tetrad there are certain points at which you can see crossing of the two chromatids are being formed. See, look at this X portion. This X portion where the chromosomes are being cross-linked to each other, and where the crossing over happens, that is referred to as the chiasmata. The points where the crossing over happens is referred to as a chiasmata. Now, correctly speaking, here. Okay, and as a result, then after that, after crossing over has been happened, what happens? The longitudinal splitting of this happens, and the process of terminalization will happen, and as a result, what happens? The tetrads get separated in the next process. Okay, and there would be what? The chromosomes which are having the maternal chromosome only, some with the maternal chromosome only, and these are the non-crossover chromatids, and here these are the what? These are the crossover chromatids, because they have already undergone what? The process of crossing over. So this is the different sequence of events you can see. Uh, in the packetine they are undergoing pairing, and chiasma formation will occur in diplotin, and after that the process of diakinesis or the terminalization happens, and the two chromosomes gets splitted with each other, and they continue with the meiosis too. So I guess you got an idea of how the process of crossing over is happening, and we'll be continuing with what are the features of chromosome crossing over, as well as the types of crossing over in the next session. Thank you for now.